Next up, we have challenges and opportunities in cannabis testing, and I'd like to welcome to the stage the panelists, and this will be moderated by Emily Richardson, Vice President of Business Development at CW Analytical Labs. Thank you.
Good afternoon, everyone. My panelists are joining me now, and I'll have them introduce themselves, starting with Tiffany.
Hi, I'm Tiffany Devitt, and I'm with Cannacraft. We are a manufacturing company up in Santa Rosa, manufacturing a full line of vape cartridges, edibles, etc. So we have a lot of user experience with test labs.
Hi, my name is Swetha Kaul. I am the Chief Scientific Officer of Cannalysis Labs. We're a state-licensed, ISO-accredited lab operating out of Orange County, and we have a lot of experience testing your products.
Hi, I'm Don Land. I'm Chief Scientific Consultant for Steep Hill Labs, a testing and research and analytics firm located in Berkeley and lots of other states. I do also have to say that I'm a professor of chemistry at UC Davis, and I'm not here representing UC Davis.
Hi, good afternoon. My name is Ian Stewart. I'm the chair of the Wilson-Elzer Cannabis Law Team. Wilson-Elzer is a national law firm. We're about 40 years old with offices in 38 cities. And we're proud to say we now have cannabis active in 14 of our offices across the country.
Hi, my name is Gary Ward. I'm the Chief Compliance Officer and Principal Scientist for PacLab Analytics. We have labs in Oregon and California. And previously, I was the administrator for Oregon, where I set up the Oregon rules and testing and lab accreditation programs.
Awesome, thank you. Well, just to get started, I personally have been in the industry almost 9 years. I've been with CW the whole time, and it's interesting to get a little nostalgic and look back at how far we've come. I really consider California the birthplace of cannabis testing. The oldest labs were born in the country. Were born in Northern California. I think it also goes to show how progressive and forward-thinking our industry has been to adopt sort of a self-policing mindset. Testing, has changed a lot from the early days. Dawn was there too, and it was very centric on cannabinoid testing. All anyone cared about was the potency, and now we're in a fully regulated, completely a total quality control regimen. So we've come a long way, and California cannabis labs are making huge strides in technological developments and in conducting research into things like naturally occurring solvents, matrix interferences in edibles and concentrates, life of pesticides in clones and generations of clones. And this is really just the beginning. And we're also creating thousands of new science and technology jobs. And it's great when we get a fresh PhD, a newly minted, chemist or biologist PhD that's coming in for an interview, and they're like, I'm so glad I don't have to go into pharma. So I think it's important to highlight sort of what we've created in this industry, and those science jobs are in our clients' companies as well. We see chief science officers and chief technical officers at manufacturers and even cultivators and distributors. So here we are. It's 2019. We made it through 2018. It was a wild ride for us all. As a laboratory operator, I know firsthand the challenges and achievements that this has been for testing laboratories, as well as the cultivators and manufacturers and distributors that we serve. And so, as we all know, California not only has some of the most rigorous testing standards that products are held accountable to, but also some of the most rigorous standards for the laboratories themselves. And so I'd like to start the first question off to our panel. What are some of the biggest challenges and accomplishments you've faced in complying with the BCC regulations?
Anyone?
Who wants to start?
Yeah, I'll start, sure. So this is from the manufacturer side. I think that one of the things we can be most proud of as an industry is California probably has the cleanest cannabis anywhere in the world.
I agree with that.
So yay for that. In fact, we did, early in 2018, we did some testing of non-cannabis products in California, and what we found was that your wine, your produce, and even baby food had higher levels of pesticides than we're allowed to have it with cannabis. Yeah. So the biggest challenge, though, is that double standard does create operational issues. So, for example, one of the problems we have as a company that does concentrates is we'll grow some flower and trim. That material will be grown organically. The pesticides will drift over from the other farms, the non-cannabis farms, and once we concentrate that and the pesticides concentrate, sometimes that drift alone can cause our product to test positive, the raw oil, at which point we have to remediate it or toss it or take other actions. The second place where you see that double standard, where cannabis is held to a much higher standard than other products, is when you're adding products or other ingredients to cannabis, like with edibles. So the biggest issue that we've recently seen now that phased-in testing kicked into effect, chocolate tends to concentrate heavy metals and pull them out of the soil. Yeah. The first thing we found was that 95% of the places where we were sourcing chocolate or tried to source it could not pass California's standards for heavy metals with regard to cannabis-infused products. So I'm not arguing for lower pesticide standards, but I sure would like to see the rest of the other industries be held to the same standards.
Yeah. You know, it's interesting that you bring up the heavy metal issue, because heavy metals is a type of test that now is pretty new to other industries too. Food and pharma only started testing it in 2016, 2017, and the chocolate industry is aware of this issue, and it's been a huge problem. And I think the way most of the other industries deal with it is when we get to a point of maturity, I think hopefully we'll get to the point where if you have COAs for everything that goes into your product, that's a good enough standard, and then you can now combine it and you have SOPs that make sure that that can be used. But it might be some time because, you know, there's still a lot of proving to be done as an industry as we grow and mature. For a testing lab, I can tell you, I mean, the biggest accomplishment for us and a lot of other testing labs is if you are ISO accredited for all of these 15 different test methods we all need to have in-house, and you were able to pivot every time they changed the regulations on us, and every time they changed action levels, and every time they changed test types, I think that's a very significant accomplishment. I come from the pharmaceutical industry, and I've worked with a lot of people in food and EPA world. Generally speaking, you have labs that specialize in one type of testing, right? You're a pesticide lab, you specialize in it, that's all you do. You're a heavy metal testing lab, you do that. We have now turned into an industry where the lab testing have had to be experts in all of them. We have to be experts in solvents, we have to be experts in pesticides, we have got to be experts in heavy metals, in microbial. Usually microbial is not even mixed with analytical testing, and now we have to be experts on that. So I think if you have survived the last few years and you have an ISO accreditation and you're state licensed, you have come a long way. I think that's something that we should be proud of. The challenges obviously are things that everyone in this room is familiar with, right? Everyone talks about consistency, standardization, you know, why do you see so much variability lab to lab? And what I would like people to understand is some of this is not in our hands. The regulations have set limits based on individual lab standards. When we pass or fail based on limits of detection of an individual lab, that means what I have at my lab as something that can pass-fail is different from what Emily would have at her lab or Don would have at his lab. And this is not by design. We do not like this regulation. We want action limits for everything. But it's something that we have to deal with. So there are challenges within the regulatory framework that are out of our control that we still have to deal with.
Yeah, I would kind of echo Swetha's comments there that one of the biggest challenges is this idea that the limit of detection is your action limit, essentially. And that is not the way to do things. They don't do that in any other industry. It's just not a good idea. You need action limits that are set, and then the LOD has to be significantly lower than that. If you're measuring and calling pass-fail at the limit of detection, you're going to get a lot of false positives
Yep.
and a lot of false negatives, and that's why you don't do that, right? So, and so somehow the Department of Pesticide Regulation and the BCC have to figure out the right way to do that and make that, work, because we have different limits of detection for every compound.
Yeah.
And so does every other lab, and they'll be different from lab to lab, and they could be different from week to week depending on the status of the instrument and so on. So that's one of the biggest challenges. Another big challenge is we have, it's getting close to 200 analytes
Yeah.
that we have to test, and no other industry requires you to get 100% of the analytes every single run, or else you have to redo the entire batch. No other industry does that. If you get a couple, you can't fail. You can't have an issue on what we call laboratory control samples, which are the positive controls that we put in the instrument to make sure that we're getting the right response, right? Several of the compounds that we have to test to degrade really rapidly.
Yep.
And they come in a huge mix that's really, really expensive, and so it's really, really expensive to recalibrate and get a fresh one of those every time. And so most industries, if you miss a particular compound, there's usually a limit on what percentage of the compounds can be a little bit low or a little bit high. But the BCC has got to be every single analyte within that acceptance criteria every single time, or you have to redo the entire batch. That's really expensive, and that costs everybody. That costs the patient ultimately, right, or the end user. So that's a big issue. And those two things, I think, are the biggest blockages to California labs being consistent,
Yeah.
right?
Actually, I'd like to just quickly add,
Yeah.
based on what you're saying, Don, I'd like to point out that this is sort of a good platform for some of our labs to just sort of get it out there that we are held to a pretty rigorous standard. It's not that we're getting away with things and that, things aren't being tested in a certain way. There's so many quality controls. I think at this point right now, 40 to 50% of all samples run in our lab are quality control
controls.
samples, which
Yep.
means we're not making money off of any
It's
of those
double
samples.
the cost for everybody.
Yeah.
Double the
Increases turnaround
cost.
time, too.
'Cause- Yeah. And so the requirements that we have to do laboratory control samples every 10 or 20 samples in a batch when this instrument is stable over dozens and dozens that's not a scientific approach.
Right.
That's a regulatory approach without any science background.
Right.
That's what that is. And it's expensive to everybody in the system, right?
Yep.
Absolutely. And I wanted to also say that a lot of people use oils, olive oil. We
Yes.
have people failing because there's pesticides in the olive oil that you buy. We have people
Yeah.
failing because there are pesticides
Similar to what you
in
said.
the essential
beeswax,
oils you
yeah.
buy,
Beeswax,
right? And
lemons.
the cannabis is clean. This is the issue. With good manufacturing processes, you have to test every input. You can't trust that the olive oil that you buy that's, FDA approved or whatever, can't trust that it's going to pass the cannabis rules, unfortunately. So.
Yep.
Sure. Well, from a liability perspective, I think that the cannabis industry is very unique in some ways. So by background, I'm a product liability attorney that worked with the nutraceutical industry for many years and had lots of contamination claims, among others. But there's some really big differences between those kind of cases and what we're seeing in cannabis. And the lack of consistency in the testing should be concerning to every licensed business in California because keep in mind that the key to defending companies, whether it's testing labs or a distributor or manufacturer, with respect to contamination or a labeling issue or whatever it is consistency and documentation. And so it's great that we got good, solid documentation from each of the labs up here, and we can validate the methods
Yep.
based on those tests. But when they differ from lab to lab and there's no set standard, even though they get ISO certification, the standards are all different. It's an opening for attorneys to make really dangerous arguments. Because keep in mind that juries inherently trust COAs. They inherently trust documents that come out of laboratories. And what really makes juries angry is when they think that documentation is wrong. And that's where we've seen huge verdicts against the dietary supplement industry over the years. And it took out a whole, remember Barry Bonds back in 2003, 2004 with Balco and all that? In the years that followed, there were thousands of lawsuits that took out a whole segment of the nutraceutical industry. And that's a very lightly regulated industry. And the other difference is in those cases, the labs typically aren't named as defendants, but in cannabis, the labs typically, and we're seeing this more and more, are being named as defendants because they are part of the they're part of the chain. They're inherent in getting that product to market. And so what that does is it sets up a very dangerous dynamic in litigation where in the nutraceutical space or food contamination cases or even pharmaceutical cases, the lab is your partner. The lab is your expert witness. The lab comes up and tells the jury how they validated the test and is a powerful ally. But if the lab is part of the lawsuit and is a co-defendant sitting with you at trial, there is a strong tendency to start pointing fingers and become adversarial. And that's very dangerous for the labs and it's very dangerous for the licensees.
Yeah.
Well, that's scary.
You're welcome.
Thanks, Gary.
So, by the way, the LCSs, the frequency of the LCSs was set back in 1981 by me at EPA, and that was because back then the instruments would drift, but they haven't updated their requirements since 81. So how many? I don't know, it's more years than I can count. Yeah, one of our problems is number one is the low levels, and we were seeing positive hits of pesticides from someone who hung up a no pest strip in his grower spot. That's how low we are looking, and it's kind of ridiculous because usually, if the person's using pesticides, it's there. I mean, it's going to be there way more than 0.01 or whatever the heck the stupid low level is. So driving these requirements down to so low is all it's doing is causing problems. And there's a lot of times with cannabis, you will see the matrix has an effect where it looks like it's there, but it's not. And when you start going to really low levels, it's very difficult to pull out these matrix effects. Whereas, in fact, we just saw an analysis last week where there was a positive hit, but when you looked at it, in fact, it was an interference from another peak. So when you start going really low, you run into a lot of problems. As far as the legal stuff, okay, so I kind of grew up at the EPA. I ran their Superfund program, and we went up against Monsanto and all those other big guys. And we developed quality control so that, and I asked the lawyers, what do we need to win? And they said, well, you have to show that it's known and documented quality, and that it has to be legally defensible. And I said, what does that mean? And he goes, well, normally we would call the analyst on the stand, but if the analyst is dead, which happened in one case, by the way, or it was like 10 years down the line when no one remembers what the heck you did, then you have to have the documentation in place to be able to stand up in court. So when I drew up the Oregon rules, in Oregon, I made the QC for Superfund the exact same QC for drinking water and cannabis. And we're trying to get California to do that. And we'll get there. There's been some big changes in the California regs since, well, I first looked at them last a year ago, January. They didn't even have an LCS.
Yeah.
And so they didn't have anything that showed that the thing worked from start to finish. Anyway, so one of our accomplishments, I guess, is getting a COA out. You would be surprised how hard it is to get. Back in December or November, trying to get a COA out was like a monumental pain in the ass because things were changing, and we knew that in 30 days it was going to make more sense, but they refused to let us do it. So we had to use the old stupid requirements. I shouldn't say that.
It's already out.
The older requirements, and we just gave up. We said we're not doing any more compliance testing until January because it was almost impossible to get a COA through. So one of our great accomplishments is now we get COAs out.
Yeah,
So,
yeah.
and that doesn't sound like much, but if you're in a lab and you're trying to get
It's a
these
lot.
results out and it keeps getting shot down, I mean, it's a pretty big thing for me.
Thank you. I know we've touched on sort of a few of these things, but I sort of wanted to highlight this. Over the years, I've definitely heard, I'm sure all of us laboratories have heard a lot of rhetoric about the lack of oversight and standardization of laboratories in California. And again, like these guys touched on, that has changed drastically. And I would like our laboratory panelists to comment on how the requirements and standards for 2019 are different, and how and what new levels of oversight and scrutiny are labs facing now that are actually a benefit to the industry. So this sort of idea of lack of standardization or lack of scrutiny or lack of oversight, I can tell you is absolutely gone. And so just if we could comment on just a few of the things that laboratories are now held to that ensures the accuracy of your data as a client of a laboratory.
So, I mean, we've come a long way for sure. There's a few things that got taken away that, you know, were good. Like we used to be able to do field sample duplicates, which meant that we could get replicate data on sets. That was in Jan 1st of 2018. That first draft, we could actually get more data on each sample. That went away, so that was a little unfortunate. But right now, moving into, especially once we move towards annual licensing, labs are required to have the ISO accreditation, which means we have the documentation that's supposed to be legally defensible. We have all of these quality control requirements, so we run several quality control samples with every batch of testing. Every failed analyte needs to get tested twice. We test in replicate, and that really has been helpful. I think, you know, it sort of eases the mind of people. Is like, how do I know when I fail it's not a false positive? Well, we have to re-prep that same sample 2 more times, and they have to be in agreement with each other before we can actually pass data out. So that should actually help ease some of the concerns with trying to get replicate data. Apart from that, just the fact that we can actually achieve some of these narrow requirements of recoveries and limits of detection, limits of quantification, and they've checked us. I mean, the BCC regularly audits, I think, all of our labs that are out here. I mean, we are required to put data packages together that are upwards of 1,500 pages, and we will still get scrutinized on that one peak that looks a little off, and, you know, go back and redo it. And this is pretty often. The data packages that we're required to send them are pretty detailed. I mean, we're talking about calibrations curves, instrumentation, analyst information, all of the quality controls, the rest of the batch data, and they look through all of this, and pretty regularly. And I know, you know, Emily has talked to me about at CW, they've already started physical audits where they're on site and they're actually comparing your SOPs to what you're doing. And so that level of oversight is pretty significant, especially when you compare it to other parts of the industry. And so that's something that, you know, we definitely would like to highlight. Yes, there are issues in the methods. And, you know, I've talked to so many of our panelists, and the truth is, you know, I think the reason this comes up is we have had some bad actors in the lab industry. We have to acknowledge that. But there are bad actors in every other sector of the industry, and no other sector of the industry is ever held responsible for the bad actors except the labs. I doubt any of you manufacturers ever got asked, you know, whenever some manufacturer had to have a recall or there was tainted pesticide products, no one ever came up to you and said, what are you doing about your products to make sure they're pesticide free? As labs, we regularly get asked, oh, there was a lab that got shut down because they had bad methods or bad data. What are you guys doing? And, you know, we look at each other and we're like, well, we're not that lab. You know, we have oversight. And, you know, that is something I would like a little more consideration on, or at least more nuance and thought about it, is that we can't control every actor in the industry. I'm not responsible for other labs, just like how you're not responsible for other businesses. But we do have some standards, and there's always room for improvement. But right now, because of all of the requirements on us, we have the most oversight of any other sect of the industry.
Anything to add?
Yeah, I was actually, earlier it wasn't like that. I mean, earlier
Yeah.
you just you were like, the state of Washington, they didn't even go to the labs. They just took their SOPs and said, oh, the labs know what they're doing. Well, they soon found out they didn't know what they're doing, and so that didn't work. In Oregon, we did an assessment of every single lab before we got accreditation. In California now, they're sending out a team, and boy, those guys are really nitpickers. I mean, they crawl through everything, and stuff that sometimes I say, are you sure this is required? But they say yes, so I say okay. But literally, the oversight has gotten a tremendous improvement over the last, 4 months, 5 months, whatever it is. And that was always my concern with California, was that ISO accreditation gets you a certain distance, but it doesn't get you all the way. There was common saying up in Oregon that said, I guess this was Intel, they said, yeah, in ISO accreditation, you can get cement life jackets and still pass ISO accreditation. And that's probably true, because the ISO accreditation is really looking at documentation. You have systems involved, but it doesn't actually
it's
look
the minimum standard.
right. But it doesn't actually look at, are you doing it right?
Or if they're
And
effective.
that's the important thing. And now California is going through and looking at our quality control
Yeah.
and looking at all our stuff. And I feel much more comfortable now that the labs, if they're passing that, they're actually in pretty good shape.
Yeah, and it's very much welcome that the level of scrutiny is being spread across the labs. And,
Yes.
and the faster that can happen, the better everybody will be.
Yeah, we actually asked them to come.
Yeah.
We
Right.
said,
We did too.
would you please come and look at us? Because
Yeah.
we've done our best now, and we may or may not be perfect, but I'm sure you guys will figure it out.
I think at one point there was a concern that, you know, certain labs were getting more scrutiny than others, and if there was enough resources with the BCC to be able to do oversight, but I do think it is more evenly distributed now, for sure.
Yeah, and I think it's important that, I mean, there are new labs getting licensed, you know, every week. So every lab is gonna go through this at some point. Now, you know, it's only a matter of time. There are obviously gonna be bad actors, just like there are in every facet of this industry. So I think, you know, we definitely as a laboratory, you see clients kind of jumping around, or they're jumping to someone with a 24-hour turnaround, or you know, and those things can sound really appealing, but you know, you also need to keep in mind, you know, is that feasible to get accurate data with these requirements that we have at this point? It was absolutely possible before, you know, regulations were in place, but it's a different world now. And that sort of brings me to my next question, and I'd love to hear from a manufacturer's perspective too. What are some things prospective clients should look out for when vetting a lab partner, and you know, both from a lab perspective and from a manufacturer perspective?
You know, that's an interesting question, because the people who know what best about what a lab shouldn't and shouldn't be doing are you guys. And obviously we have scientists on our staff, but those scientists aren't the sort of scientists that necessarily understand all of the idiosyncrasies and everything that you have to do this sort of thing right. So where we end up, the way we end up making that decision is really trial and error. Meaning we send out samples, we have them tested in another lab. So initially when we were doing the vetting process, we were testing our products at multiple labs, right? Or we would send the same sample in under 2 different names to a lab in order to find out, are they getting the same results from one time to the next? Are their results consistent with this lab or that lab? Now, that said, one of the things that does happen with manufacturers is you do end up kind of getting locked into a specific lab. And what I mean by that is, and this gets to all the things these guys have been saying about standardization, we obviously do a lot of in-process testing, right? Because we need to, before we put oil into a vape cartridge or an edible, we need to know it's clean, right? Otherwise, we're wasting our time and money. So if we don't do that in-process testing with the same lab that we do the finished, that our distributor uses to do the finished good testing, we can end up with different results, in which case we've labeled our product incorrectly. And then it gets kicked back and rejected. So it's a process of trial and error. B, you have to have your own methods of validating it from one lab to the next. And B, you better hope you've made a good choice, since you're gonna have to use that same lab through the entire manufacturing process.
Yeah. I would definitely like to address this whole idea, because I know this is very common practice where people send the same sample out to different labs, and then they're like, well, the results are different. And you know, one of the things I'm sure my colleagues would agree with is they probably are all right.
Yeah.
They're all right, they're all different, and they're all correct, because the truth is we don't have the best homogenization practices. We don't have a lot of standardization, even in the lab, on how to deal with the different kind of matrices that are thrown our way, and how to deal with mixing them and making sure they're homogeneous. So there are issues there. But when you send a small retain out of one part of your, you know, few liters batch to different labs, it's not the same as doing a compliance testing. And I'd like to, sort of very simply, in compliance testing, we're supposed to do representative sampling, which essentially means is if you can imagine your container, you have to break it up into a grid system, into a grid, and then number the grid and randomly select using a randomizer different parts of the grid. Now you pick those, you mix them up together, and that's your representative sampling. Very few manufacturers actually do that in order to send that as a retain. Usually they're just picking one off the top and then sending it off.
Yeah, we do that.
Yeah, and I mean, I'm so glad
For
you're
all the
doing it.
reasons you say.
And you probably see a lot more consistency in data once you start doing something like that. And the other issue, obviously, is something that we as labs, I know everyone here has fought against, is the batch sizes. I know, there's a lot of pushback about the cost of testing, and, one thing I definitely want to make clear is like the labs had nothing to do with the regulations. We wanted to change a lot of things, but we didn't. And we want a thriving market because that means we thrive. That's just the end. That's the truth of it. But if you want accurate data, you need replicates. You need more samples. You need randomization like this. And there's always this kind of push and pull with, are you willing to do replicates? No, it costs too much, so we're going to take it away. And the same kind of argument happened with batch sizing. Our batch sizing is 50-pound batches. We have the largest batch size of any other state in the nation. And it's just very hard to get representative sampling out of such a big batch. And I know a lot of people who have experienced this have literally gone to smaller batch sizes because they see more consistent data with smaller batch sizes. And this idea of trying to get different parts of something that's inherently not homogeneous, it's a challenge, and I know we all deal with it on a constant basis.
Yeah. And just because something looks homogeneous doesn't necessarily mean that it is. I think that's a big misconception.
Can I make a comment? And I don't, all the scientists up here are sort of intimidating, but from my perspective,
We're scared of lawyers.
yeah, good. You should be. But when you think about it, I hope Gary and Don don't shift me under the table here. But for customers who are vetting labs, I think obviously reputation is important, and that's a good thing here. But the bad thing these guys aren't going to want to hear is you gotta think about the risk transfer, right? You got to think about the indemnity and the AI, the additional insured status, potentially, in the chain of distribution. I know different companies do it different ways, but I will say that the insurance industry as a whole is very concerned about the testing aspect of cannabis, not because any of these companies are doing it wrong, but because of the potential for when things do go wrong. The good news is that what we haven't seen is the predicted wave that was being predicted 2-3 years ago of bodily injury product liability cases. I mean, these are the cases where you've got somebody who takes a product and there's an injury or property damage caused by the consumption. What we do see are sort of what we term the product tangential cases, right? So it's the recalls and the contamination claims and the consumer class actions, which are now starting to be filed because of the consumer protection statutes we have here in California, which are very ugly and which contain really nasty statutory remedies, including disgorgement of profits. They can seek accounting, so you can go back and go through the books for the last 2 years and disgorge all those sales. Punitive damages, et cetera. And so the reason why the insurance industry is so concerned is because they've got all this data for other industries. So we talked about nutraceuticals a minute ago. And when you think about it, what happens when you read the news and, there's a report that the proprietary blend in some product that's on the shelves at GNC turns out not to be really the blend. It's not accurate, right? Well, there's sort of a collective shrug. People go, we kind of expected that, right? I mean, it's not good, but nobody's screaming about it. But what happens when there's a problem with baby food, right? What happens when there's a problem with dog food, right? I mean, we care more about our dogs than we do about what's being sold, at GNC. Well, I hate to say this, but the public thinks of cannabis like dog food. I mean, I think that's what's going to happen, and what in terms of they're going to be mad, and what risk management industry as a whole is really fearful of is this thing called the reptile theory. I don't know if some people in the room have heard about the reptile theory, but that's what plaintiffs' lawyers have been using over the last 10 years or so to jack up these huge verdicts that are way out of proportion with the actual economic or even the non-economic damages in the case. And the way they do it is they play on cognitive bias and instinctive fears by taking the allegations outside the context of the case itself and putting it out in the public at large. So saying it's not just, when Steep Hills did this, it's not just you that they harmed, but they're harming, it could have been your children, it could have been your mother, it could have been your neighbor. And they use this fear, and fear and anger are very similar in the brain, and when a jury gets angry, that's what drives these verdicts. And I think that's why the insurance industry is sort of concerned. We haven't seen it yet, but that's the concern amongst the underwriters and the risk managers that are looking at this industry saying, should we jump in?
Well, one of the problems, the labs, when they do an analysis, they've got enough QC, so it's literally the lab analysis is as tight as Tylenol. I mean, our spread in our analysis is the same as Tylenol. The problem is that we've got a plant with parts and goo and sticky crap and all that other stuff. And
Those are
as
technical terms.
Swetha pointed out, 50 pounds. 50 pounds. You know how big 50 pounds is? I mean, it's unbelievable. And trying to get a sample out of a 50-pound batch is ridiculous. Oregon's is 15 pounds, and we did that with a statistical study that shows that when you go more than 15 pounds, you get differences just because it's a different size. And what we need to do is, if you're going to do 50 pounds, you're going to have to do replicates.
Yeah.
You should do at least 2 in different places, because if a client takes a sample and then sends it to you and takes another sample, the odds of that sample being the same are like minuscule. I mean, it's almost impossible to get the same thing, because you can homogenize it, you can do all that other stuff, but then you're taking 0.2 of a gram, which means you've got this big clump of glue and you're taking this little bitty bit, and that's what your pesticides is. So, it's not that the labs are not doing things properly, it's that the sample it's inherently a crappy sample. I mean, literally, when I was in Superfund, we would look at sludges and wastes and barrels, and they were easier to analyze than marijuana, because marijuana is such a mess. I mean, you got leaves and chromosomes and stems and roots and all kinds of crap, and you try to grind it up, but when you're done, you get this ball of goo, and it's like, how the hell do I get, without analyzing the whole friggin' thing, how do I get a sample out of it?
Yeah.
So the problem is that it's an inherently bad matrix. And the worst thing California did was take out the duplicate sample.
Yeah.
Because now you don't know that if I analyzed it a second time from a different place, would I get something close? So our recommendation, and CCIA's recommendation, has been put the duplicates back in. I know you don't like to do that, but that's the only way you're going to find out whether or not the sample itself is the problem.
What are the batch sizes in other industries? Like for pharmaceutical, for example, like what's the batch size for Sudafed?
It's defined by production lot, and so in pharmaceutical you have one API with a bunch of fillers, and it's a known product. Everything that goes into the product is manufactured, and so if you have a production
what about
batch,
like strawberries or something, or agricultural product where the pesticide could be sprayed on it in a variable way?
Sure.
You would have similar issues. So I'm
Right.
just wondering what's.
It's honestly, each industry has their own lobbying group that works on it separately. So you'll see everything from 5 pound to 20 pound batches. Yeah. It's a wide range, but it's all based on what Gary said, which is statistical analysis. And you basically do replicate testing and figure out at what point is this redundant. And so we never did that for 50 pounds. We don't have, I know we've done that study and showed that it's very hard to get consistency with 50 pounds, but this is where the regulations is. So I think most of us always recommend, if possible, if you want a better chance of passing, you should split up that batch.
Well, it's also interesting because there's food and there's drugs, right? And in this industry, we're being regulated as basically a food and a drug.
Yeah.
And there's inherent inconsistencies with those types of regulations, and that we're now all caught in the middle of. And sort of to go off what Gary was last talking about, I'd really like to have the panelists comment on what sort of advice or information would you give your clients to generate more accurate data, because as we've sort of been talking about, accurate data is not a one-way street. It's not just the lab. The whoever's producing the product or taking the sample plays a vital role in how accurate and reliable that data is. And so in order to create better relationships between laboratories and our clients, to really see it as a partnership rather than what it can sometimes get to this sort of antagonistic this antagonistic level. And I know as a laboratory operator, we hate that. We never want to see that. We want our clients to see us as partners. We want them to trust us. But so what, from a laboratory perspective, how could your clients help you help them in terms of generating better, more accurate, more consistent data?
I can go. Well, I think the first thing would be, please don't threaten your labs. I mean, it gets really old when you threaten labs every time you fail, which is a very common thing that I think we've all experienced. Is every lab always experiences this. Is you have a failed result and you have a client that threatens to never test with you ever again because you failed them, and they're very mad at you. And it's like, you can't take it personally. If you want an accuracy in a lab, and we'll stand up to this, you cannot have that happen. And it unfortunately does happen very frequently. There's a lot of pressure and a lot of threats, and I'm not surprised that there are actors that fall for it and give into it. But the pressure is not coming internally, it's coming externally. And the market forces are kind of forcing these labs that already are struggling with margins and regulations and equipment to think, well, maybe if I don't want to lose this one major client that I have to give into it. And so, let's not do that. The other thing would also be, lab shopping is a very similar thing. It's externally driven. We don't want to see lab shopping. I honestly, when I see clients that come to me with R&D tests from another lab, I'll tell them to go back to that same lab. We want to see consistency in data, but if you just keep hunting around for the best possible data to make sense for you, it's not creating a good information or reputation for us as a whole. The other thing, obviously, we've talked a little bit about the homogenization, the batch sizing. If you can possibly test in replicates, try and get multiple data points. I think that's all, things that would be advisable. But there are a few things that I know, just very simplistically, because we've talked about this, where, when people are formulating things and manufacturers are formulating things, we often hear of people saying, I mix this volume with this weight, and it's like, you can't do that. And the reason is that generally volume is one of the most variable things based on temperature, density, things like that. Your volume is going to vary when you're measuring it. So we always recommend for everyone to do it by weight. You always mix your formulations by weight. It's more accurate, it's more consistent. Most of the people who've been around for a while have already figured that out. But these are small things that do help get more consistency in the result. Also, we're not dealing with an API. Like when I talk about API, this is a pharmaceutical. It's the active ingredient. We don't have a single one. We have something that is varied. It's a mix. It's with, like Gary said, it's just a bunch of goo, and with a lot of different things in it. And it's hard from lot to lot to get consistency, because every lot of that oil or distillate you're using is completely different from the previous lot. So your math might have worked for that previous lot, you have to redo that math again for the new lot. And it's a constant process, it's an iterative process, and you have to keep improving on it. That's a few I can think of. I'm sure you guys have a few more.
No, I would just expound on one of I think you covered it pretty good. The one thing that I would expound upon is that just because something is a liquid does not mean it's homogeneous.
Yes.
Viscous liquids are incredibly difficult to mix. Look in the food industry literature. It's incredibly difficult to take these viscous oils and try to mix them together or put other ingredients in and get something that's homogeneous. You would not believe how long it takes to mix those viscous liquids to get them to a reasonable point of homogeneity. And this is the biggest problem we see, whether it's the lab shopping or sending multiple samples, is you can't just take one sample. You can't.
And just for perspective,
and even taking multiple samples from this very inhomogeneous liquid, if you can look at it and you can tip it and you see these little waves in it, that is not a homogeneous sample. All liquids are not homogeneous.
Think about your ketchup in your fridge. When you dip over your ketchup, water and stuff comes out,
Yeah.
and the rest of it is kind of goo. So, I mean, that's ketchup.
Just shake it, Gary.
Yeah, I tried to shake it. I think the stuff is thicker at the bottom.
Well, and just going off what Don said, just so people get a little perspective, in some of the industries that deal with viscosity, like chocolate or honey, they have protocols that have it mixing for days at very, very, temperature controlled, at very high RPMs in a big, stainless steel container. And I know some manufacturers understand that because they've done studies on that, but a lot of people, we've probably all heard of the person that said, well, I mixed it with my wooden spoon and it seemed fine
Yeah.
to me.
The one on the drill.
The EPA has a test called EP toxicity, TCLP, and it's to analyze garbage dumps from hazardous waste and acid rain. And you tumble that for 24 hours
Yeah.
before you take a test. I mean, it's extremely long, and it has to be a certain temperature. Temperature can't change. It's subject to all kinds of stuff.
We
But
have
one-
a.
But one of the things that you said, well, what do you look for when you're looking for a lab? And when I first took over the Oregon lab system, the medicinal was going before I got to it. And then what there was is I found that there was a list, I literally saw it, a list of labs, and next to it were THC values, and people would go for the highest THC value. Well, to be perfectly honest, I would stay away from the top THC value because it's almost always inflated.
Yeah.
So you can't really go, I hear people say, well, I get 22% out of this. I'm going, yeah, but do you want the truth or do you want something fancy for your label? So you can't necessarily go by that. You really, for looking at a lab, you should look at their reputation, their quality control, how good the reports are, and and go by that. But then it's still a hit and miss. You still try them and do it because there's no easy way of doing it.
I just wanted to.
Oh, I was just going to say, I'm so sorry that you guys have been threatened by manufacturers, and we have not threatened our lab. And I have to say, I'm shocked because lab services are so in demand
Yeah.
that the last thing I would want to do is make my lab mad, so they put us at the bottom of the testing queue. But
Well,
wow,
it's
that's
like-
terrible.
It's like
It's a
pissing
very
off
common practice.
It's like pissing off the waiter. You're just going to take it back
Exactly.
and spit in it.
Don't say that, Gary.
I wouldn't say as a lab we would do that, but I'm saying we pretty much say, look, this is the number. If you don't like it, then go someplace else.
Yeah.
I do want to add in a couple comments to how to generate better data. We see a lot of clients that are running experiments, but we're completely blind to the experiment, the desired outcome, what they're even looking to investigate. So, and a lot of people have the mistaken belief that that's necessary to withhold that from your lab because that's helping them cheat or that's biasing them. So I can't stress enough how important it is if you're trying to run some sort of experiment or investigation into an internal process or a formulation or something like that, involve your lab. And if you don't trust your lab, then you shouldn't be working with them. So, sending blind samples, people will spike things with really high levels of pesticides and it will completely screw up a run on our machines. Where if we knew that something was going to come in super concentrated, there would be blanks run before and after it, things like that. So these sort of gotcha games that get played really don't help you as a manufacturer, distributor, whatever you are. So find a lab that you trust and work with them and really involve them when you're creating an experiment or you're deciding whether to investigate a hypothesis you have or a homogeneity study or whatever it is you want to do, because again it's help you get better data. So just wanted to throw that in there as well.
Yeah, no, it's, the more information you can give us, the better we can do it. I remember we were working with a cookie manufacturer, and we were finding that he had jelly in top of the dough, and the jelly was the part that had all the THC in it. And so we worked with him to have get his process down so he could get the dose he needed. And that's what you need. It really should be a
Partnership.
partnership. It's we are trying to help you get your product out and
Yeah.
safe.
And I think that's also true for the for issues involving remediation and
Yes.
retesting. So when there's a recall or some sort of failure, we found that the best outcomes are the ones where there's collaborative effort and not confrontational effort.
Yeah.
So.
Awesome. So So we have about 10 minutes left here, and I sort of just want to end this by looking to the future a little bit. And I'd love to hear what all the panelists have to say about what changes in testing regulations would you like to see? What will we see? What does the next 5 years look like, as opposed to we're in the trenches right now? What is it going to look like? What should it look like a couple years down the road?
Okay, well, one of my pet peeves is the micro. California is looking for E. coli, Salmonella, and Aspergillus. Well, the trouble is we have seen product that has passed for those species, but is covered with mold.
Powdery mildew.
And UC Davis did a study and found, when they went to dispensaries, they found other funguses like mucor and cryptococcus in the cannabis. And I think last year, was it last year or late last year, some gentleman died because he died from the mucor fungus. So one of the things that I'm trying to get California to do is put in a overall yeast and mold. In other words, look at a total yeast and mold so you get all the different species, and if it's in there, then you worry about what it is. But we're trying to get them to put in this test that will kind of cover the bases across the board, because if it's not Aspergillus, if it's Cryptococcus or something, you're going to miss it.
Yeah.
And so I think now Nevada kind of had the right idea. They put it in place, but they require the plate technology, which is like 100 years old and useless, instead of the qPCR, which is quick and cheap. So they had the right idea, they just implemented it wrong. But that's, I think, in the future, that's, I think, where we're gonna go as far as micro is looking at an overall mold content, fungus content, and then if you find stuff, then you go in and find out what it is.
Yeah.
I think one of the changes that's coming, which I am going to welcome, and probably the BCC and regulators will not. I'm actually interested to hear Gary. The new ISO, right? The ISO 2015 requires. Let's say that the action limit for a pesticide is 100 parts per billion, and we tested at 110. But our measurement error is, like everybody, about plus or minus 10, 20%. So that's what's called an indeterminate result. So technically, it's above the limit, but it's within our uncertainty, so that it could actually be slightly below the limit. And the new ISO rules that came out in 2015 and are not required yet, but kind of are, says that an indeterminate error is reported as such. And I don't know if there's anybody from the BCC here or not, but that's coming.
Yeah.
So we actually had a very similar situation in a case about 5 years ago involving a large shipment of beta-carotene that came out of India to be put into children's vitamins, and there was I forgot what the standard was, but it was a very, very strict threshold for any kind of microbial contamination, and so same thing where it was reported as negative, but it was right on the edge in terms of the detection limits. And the problem we had in that case was we had great documentation. We validated the results. We had a retained sample that was tested, and that was clean. So we were sure that the contamination that was seen happened down the road, once it got to the manufacturer and was put into the final product. But the product came out of India, and we had perception issues. And I think that to some extent, those perception issues are true for this industry as well. And so there's going to be very little margin of error in terms of an attorney trying to defend these suits compared to a large food lab that works with like a Nestle. I just think that there's a perception issue, and so that is not an insignificant problem that we're going to have to overcome.
Hey, what's wrong with India?
Nothing. They were clean.
No, I know.
I also think that in the future we'll be doing scans. So instead of picking 4 metals, with ICP-MS, you just scan 20 of them, and it's the same amount of time almost, and that way we're covering all the bases. And with GCMS, you can do scans and look for all kinds of solvents,
the unknowns,
and all kinds of terpenes, and it doesn't take much more time. So I think we're going to go more towards scanning, and then finding the things that we weren't looking for necessarily, but being able to report them.
I mean, I think we're come a long way. I think, the ISO requirements are going to hit in for annual licensing, so we are going to see a drop off, I think. I think there'll be fewer labs at the end of this year than we saw in the beginning of the year. And it might not be a bad thing, because the ones that survive are the ones that are, trying to do it right, that have the quality systems and that can survive. As far as the actual methods, I think we are going to start seeing trends, because there's such a big need in this industry for that turnaround time and the push towards, everything quicker right now. And even though some of it, we'll probably push back as labs, it is driving us towards innovating as far as automation and robotics. And I think you're going to see some really cool things where the cannabis industry is actually going to drive some cutting edge science and technology research, and I think we're going to start seeing that come out in the next few years just because of that combination. Yeah, hopefully. It's not here yet, but I think we are going to start seeing that trend. I think what Gary had spoke to, right now we have the specific list of analytes, and I know Ian had mentioned about this liability issue with, what if something's dangerous in there that you don't even know? I think legally, because we have the regulatory system with a set list of analytes, that's our defense, right? We're looking for these analytes. But I think there will be interest to see what are the things that we don't know in there, and that's what Gary's referring to. The scanning method is essentially an unknown, like looking for unknowns in our product. So looking for unknown pesticides or looking for unknown contaminants. I think that's going to be more and more interesting. And again, it's some of these pressures of the industry are going to drive the labs that do survive to do a lot more innovation on the technology front, which I think is a good thing.
Yeah, that's a good segue to what I wanted to say. Two points about where I'd like to see the testing lab industry go. One is, I forgot to mention that I'm the Chief Compliance Officer at Cannacraft, and I basically say the same thing to the agencies in 1,000 different ways. And the thing I say is, please treat us like you treat every other industry. And one of the things the scientists on the panel have described is the hugely different standards that they are held to as a cannabis lab relative to someone testing other products. And that has to change in order for the small number of labs to support an ever-growing industry, right?
Thank you.
The other reason to kind of rationalize that is so that they can take the substantial brain power and intellectual power that resides in the testing lab side of the business and leverage that for the industry as a whole. So my colleague here was alluding to the opportunities to do research. I have a real desire to have our lab community provide clear direction on how different strains relate to each other. And there have been some different organizations who put together phyllo trees and whatnot, but I'd like to be able to go to them and say, hey, we're low on X strain, Granddaddy Perps, please tell me what strain has a cannabinoid and a terpene profile which most closely maps to that. I would have to say that our recreational and our customers and our patients would also benefit from that data. And that furthermore, to the extent that they can be a repository for that, the knowledge about the deep biological diversity in the cannabis plant, they will provide kind of buffering against big ag who's coming into California and trying to push us in the direction of a monoculture. Yeah. And if we know anything about the cannabis plant, it's that there's enormous diversity, and we've just barely scratched the surface as far as understanding what's valuable in that plant. I mean, a few years back, no one had even heard of CBD, and now it's like the it thing. But there are in all likelihood a number of hundreds of different minor cannabinoids and terpenes and flavonoids and whatnot, that if we don't identify them and preserve them, they will not be there in the supply chain down the road once we discover what they're good for. So I would love to see our labs basically be sort of the repository of what should be a shared knowledge base.
Can I just add that I know, just talking to all my colleagues here, that I love that point, and I know, talking to a lot of my colleagues on this panel, that we all love the R&D part of our jobs. There's a lot of parts that, I love looking at the analytics of how strains match up to different content. We have a lot of our side projects, but the truth is the pressures of the industry as far as the turnaround time and demand of samples, most of our time ends up going into production, and which is fine. And that's what pays the bills, and that's what's important, and that's what we want to be efficient at. That's what's going to drive the innovation. But we would love to have the time and resources to be able to put into some of these R&D.
standards for those things too. We
Yes.
can't measure something if we don't have a standard.
Right,
for it.
but-
So our standard companies need to keep up with that,
Yeah.
to be producing pharmaceutical grade standards that we can then investigate more
Exactly.
cannabinoids and more terpenes. We can't just come up with that,
Right, but I just want to hold one point, which is the tragedy of the cannabis industry being overregulated and overtaxed is the first thing we are losing is the resources and ability to pay attention to the innovation.
Yes. No, that's very
We'll
true.
end there.
Yeah.
Thank you. That was a
We
great
completely
point,
agree.
Well, thanks everyone, and thanks to our panelists, and